appointment is PhD from Fudan University Institute of Biomedical Science. And his postdoc training from University of Texas Southwestern Medical Center. He joined Huashan Hospital Institute for Translational Research of Fudan University in 2019. So his research interests include the role of natural immunity and metabolic regulation in the occurrence and development of brain disease. Since 2011, he has published 17 academic papers in top journals, including Nature Cancer Cell, which is a cover paper, and Nature Metabolism Extra for etc. For 12 of which he was the first corresponding author. These papers have been cited more than 6,000 times. 5 of his papers have been rated as highly cited paper by Web of Science. He's currently a member of the Neuro-Oncology branch of the Chinese Neuroscience Society, a member of the Neuroglia branch of the Chinese Society of Neuroscience, a young director of the Metabolic Biology Society of the Biophysical Society of China, and the secretary of the Brain Disease branch of the Shanghai Neuroscience Society. We're so excited to have them with us, and we also are very lucky and excited to have a nice lineup of panelists with us. So joining us for the panel discussion are Dr. Shi Yufeng, and Dr. Shi is an expert in cell and cancer biology. Currently, the focus of Shi Lab is exploring the molecular mechanism underlying the high dependence of the mitochondria function by glioma stem cells and identifying druggable mitochondria targeting these deadly diseases. He, as the first author, has published multiple papers in Cell, Nature, Cell Immunology, and Molecular Cell Biology with total impact factor more than 100. Professor Shi has an international patent for an anti-tumor small molecule. He was the co-investigator in a funding granted by Memorial Sloan Kettering Cancer Center for Drug Development, which is also very highly esteemed center in US, and also participating in 2 NIH and 3 state research grants. He received multiple awards, including one of the 100 best PhD theses in China, one of the 50 excellent doctoral thesis in Chinese Academy of Science, among many others. And as currently professor at the University School of Medicine. We are also very happy to have members from GOMIX today with us. We have a postdoctoral researcher, Sun Rui, and she received her bachelor and master degree in pharmacology from China Pharmaceutical University and PhD degree in biology from Westlake University, Juilliard University Joint Training Program. During her PhD, her research interests are precise molecular characterization, the prognosis determination of various diseases based on proteomics. In July 2022, she joined the GOMIX lab at Wesleyan University as a postdoc researcher, focusing on interference-based functional proteomics research. Next, also a student who is at GOMIX lab, and her name is Xiao Qi. So Qi received her training of pharmacy in HuB Medical University and obtained a master's degree in biochemistry and molecular biology in Dalian Medical School. In 2021, she joined the GOMIX lab as a PhD student. Last but not least, we're very happy to have Dr. Guo Tiannan with us, who is the PI at Westlake University and PI of the GOMIX lab. So his current research focuses on developing proteomics technologies for precision medicine, exploring diagnostic biomarkers for thyroid cancers, and understudied proteins. So without further ado, let's welcome Dr. Wes sharing his talk. And his talk title today is Novel Insights and Approaches Against Glioblastoma. Welcome.
Okay, hello everybody. Can you see my screen?
Yes.
Okay, great.
Yes.
Yeah, welcome from my side. It's a great pleasure to speak here and presenting you our work where we used proteomics to gain novel insights into glioblastoma and lay the basis for novel therapeutic approaches. So first, a few introductory words. So what is glioblastoma? it's the most common and most aggressive primary brain tumor in adults. Here in the lower left, you see a typical MRI scan from a patient with glioblastoma treated in our department. You see the contrast-enhancing tumor that arises and grows in the brain. It's never metastasizing. It's really a localized problem of the brain. But it's a very devastating disease and currently probably one of the most challenging cancer types. It's treated with a combination of surgery followed by chemotherapy with a drug that is called TMZ or temozolomide. This is an alkylating agent, as well as radiotherapy. But despite this combination therapy, the outcome of patients with glioblastoma is poor. So the median overall survival is in the range of 16 months. And as you can see here in the lower right, the survival has only slightly improved over the last decades, basically. So there is really an urgent need for a better disease understanding and also novel therapies. Recent advances in technologies, especially transcriptomics and genomics, led to a molecular characterization of the disease. And this slide just shows the current knowledge based on transcriptomics. And you can see on the left that bulk sequencing studies from bulk tissues that used RNA-seq basically led to the discovery of 3 subtypes, transcriptional subtypes of glioblastoma. They have different names: mesenchymal, classical, and proneural. The details are not important here, but basically on a transcriptional level, there are 3 disease subtypes. So far, they are not relevant in the clinic. Then, of course, with emerging single-cell technologies, single-cell RNA sequencing led to the discovery of 4 distinct cell states of glioma cells. They are all present in the tumor, and actually it's a very dynamic process. Also, this has no clinical implication so far. And of course, there are many other studies that try to understand the microenvironment, and this is really an emerging topic. So this slide summarizes the trajectory of a patient with glioblastoma in the upper part. So basically, for almost any patient, it's always the same story. So this patient has newly diagnosed glioblastoma, so the tumor is diagnosed. Then the patient undergoes surgery, followed by combination of chemotherapy and radiotherapy. But as I mentioned before, the tumor almost always recurs, so there's always recurrence. And then there are different treatment options at recurrence. I hear an echo, so probably somebody...
us too. We can also hear the echo for some reason.
Okay, now it's gone. Okay,
yep,
great.
Better.
So basically the tumor always recurs, and then at recurrence, we have no standard of care. So basically here, this is the room for experimental treatments. Patients can have other chemotherapies, second-line irradiation, surgery, and so on. But eventually, this is a lethal disease, so almost every patient dies. And there are 2 main challenges for glioblastoma. They are highlighted in the bottom. First is we need a better understanding of the evolution of the disease from newly diagnosis to recurrence. And for this, there was an international consortium formed. It has the name GLASS, or Glioma Longitudinal Analysis Consortium. This has been founded by Rolf Verhaag in the US. It's now expanding basically on an international level. I'm part of this consortium, and the goal here is to understand the evolution of the disease by profiling matched glioblastoma samples that are obtained at newly diagnosis and at recurrence after surgery and chemoradiotherapy. So far within this consortium, mainly genomics and transcriptomics has been used. The second main challenge for glioblastoma is, as I mentioned before, there's an urgent need for novel treatment options. And in this webinar, I will present you the 2 aspects, and we start first with a better characterization of the disease evolution. So in this GLASS consortium, so far genomics and transcriptomics have been used. And this slide just summarizes the findings. So if we focus on the left side, first genomics. So basically, in large consortia, matched paired glioblastoma samples have been profiled, where from each patient, the tissue was available at initial diagnosis and at recurrence after standard of care therapy. And to make a long story short, basically what they found is that there are mutations, so driver of the disease, that are maintained throughout, and there is not really a clonal selection. So there are some different clones that evolve, but overall, primary and recurrent glioblastomas are quite similar, indicating that our treatment really causes a poor selection pressure on the disease. And on the right, you see the findings with transcriptomics. And basically, I mentioned this transcriptional subtypes before that have no clinical relevance at the moment. But essentially, what people have been found overall on a transcriptomic level, primary and recurrent glioblastoma are quite similar. There are some tumors that switch in the transcriptional subtype, but as mentioned before, this has no relevance for the treatment of the patients so far. So to sum up the current knowledge, basically, there is only a minimal selection pressure in glioblastoma from initial diagnosis to recurrence after standard of care therapy. And of course, an ongoing question was that has never been really addressed is how is this on a protein level? Because eventually proteins are the targets for drugs, and of course they determine the phenotype. So we addressed this together actually with a team from the GUOmics lab and collected 2 independent patient cohorts of matched glioblastoma samples that were obtained at initial diagnosis and at recurrence after standard of care chemoradiation. So on the left, you see an overview of the cohorts. They were independent, collected, actually even from different countries. And also there were different samples. So in cohort 1, we had punches from the tumor regions that were pulled together. And in cohort 2, we had basically whole slide sections. And then we basically used PCT swaths to get the protein profiles of these samples. On the right, you see just the distribution of age and the survival across 2 cohorts and also the gender. The message is there are no systematic differences between these 2 cohorts. So no bias in this manner. This data shows the unsupervised hierarchical clustering and principal component analysis of the global proteome of these samples. And the clinical annotations are annotated here. And basically, the initial tumors are gray, the recurrent tumors are black. And what you see, there are no distinct clusters. So it's really a mix-up. And this you can also see on the principal component analysis. So essentially, initial and recurrent glioblastoma samples do not form distinct clusters, meaning, again, that there is no strong selection bias and that the initial and recurrent disease is quite comparable, which is in line with the previous genomics and transcriptomics data. And then we basically focused on proteins that are differentially regulated. So on the left, you basically see the dot plot that takes into account the 2 independent cohorts. And here, basically, we display in red proteins that are 2-fold upregulated at recurrence, and in blue, proteins that are 2-fold downregulated in comparison to the initial disease. And then we wanted to focus basically on the upregulated proteins because we assumed they are important for tumor progression and that they basically are also targets for novel strategies. And what we did is a careful literature research. We could confirm that a lot of these proteins that are actually upregulated at recurrence seem to have a role in tumor progression based on prior literature. But we also found proteins that have actually never been studied before in the context of glioblastoma. And I took a selection of these proteins here on the right. So essentially, we had 5 proteins that have been really understudied before, and their role for glioblastoma was not clear. What we did is, of course, we started with validation using antibody-based immunohistochemistry. So we took, again, independent matched patient samples obtained at initial diagnosis and at recurrence after standard of care therapy. And then we stained these sections, quantified the stainings, and could validate out of all the proteins that we had, essentially 3. And this is a protein that has the name BCAS1. It's a myelin-associated protein. Then a protein named INF2, or inverted forming 2. This is a protein that is important for actin polymerization. And then a protein called FBOX2. This is a protein that is involved in the complex for ubiquitination of proteins. As you can see, another protein, PRPS2, could not be confirmed in this independent validation. So we basically continued and focused on these 3 proteins, BCAS1, INF2, and FBOX2, to understand their role in glioblastoma. And what we did is we generated basically knockout cell lines. So we used human patient-derived glioblastoma cell lines and generated knockouts using CRISPR-Cas9. And then we studied the phenotype of these knockouts using different assays that assess the proliferation of cells, the migration, the invasion, the sensitivity to irradiation and chemotherapy. And to make a long story short, essentially, we did not observe any in vitro phenotype upon knockout of these 3 genes, so BCAS1, FBOX2, and INF2. So in vitro, no phenotype. Then, surprisingly, we implanted these cells, so the wild type cells that have the genes and proteins, and the knockout cells that basically lack them, and surprisingly observed a very strong phenotype in this in vivo context. So in the upper left, you see survival data, Kaplan-Meier curves, for 2 independent orthotopic glioma mouse models named LN229 and ZH161. The details do not matter, but essentially, especially for LN229, you see that the knockouts for FBOX2, these green curves, they really conferred a survival benefit compared to, in gray, the wild type parental cells. We also observed this for ZH161. We had also phenotypes for the other proteins, but most consistently was the phenotype for FBOX2. So we then basically focused on FBOX2 and try to further investigate this tissue-dependent context in a more relevant setting. What we then did, it's shown actually in the lower left. We used an organotypic slice model. So from patients that had actually a surgery for glioblastoma or other neurological diseases, we obtained the healthy cortex of these patients and made sections, brain sections out of this healthy cortex. And then we injected in these sections our wild-type parental cells or the knockout cells. And then we observed the growth in these brain sections over time. And basically what you can see is there was a striking difference in the morphology of the wild-type parental cells or the FBOX knockout cells. And when we quantify this, it's pretty clear that the FBOX knockout cells could not invade into the brain parenchyma comparable to the wild-type parental cells. So somehow the invasion pattern into the brain is impaired upon FBOX2 knockout. And then we went basically to another data set where we had MRI images, so imaging data from patients with glioblastoma, and matched RNA sequencing data. And we wanted to understand in these patients if tumors that had a high FBOX2 expression compared to tumors that had low FBOX2 expression have a different morphology, basically. And what we then quantified is the so-called sphericity. So if the tumors rather grew as a ball or as an infiltrating mass that really infiltrates the brain. And as you can see, the more FBOX expression we have, the more infiltrative the tumor is, which is basically in line with the organotypic slice model that we have. Here in the immunofluorescence images, you see that if we lack FBOX, we have a more roundish growth. If we have FBOX, we have a more invasive growth into the brain parenchyma of the glioma cells. So to better understand this phenomenon, we then went to spatial transcriptomics data. And in this spatial transcriptomics data from patients with glioblastoma, we focused on glioma cells by copy number operation, and then basically investigated FBOX2 expression in the glioma cells in a spatial context. And in this data, we basically always have a tumor is defined by a tumor core and in so-called infiltration zone. So at the infiltration zone, the glioma cells really invade the brain. If we now overlay on top the FBOX2 expression, you basically see that FBOX is mainly expressed in the glioma cells that are really infiltrating into the brain parenchyma. And we showed this for several samples. In addition, we also confirmed that at recurrence, it's upregulated, which is in line with our proteomic data. And then if we try to understand signatures of FBOX high expressing glioma cells, again, from different data sets that either used bulk RNA sequencing or single cell RNA sequencing data, and here this, let's say, large orange balls, you see that glioma cells that have a high expression of FBOX2, they have actually neuronal phenotypes. So they have like neuronal progenitor cells. They actually have this neuronal phenotype, and they have a lot of genes or molecules that are involved in synaptic processes. So basically, they form synapses for the interaction and communication with neighboring cells. And to confirm this, we stained in the organotypic slice sections from patients for synaptic proteins, and PSD95 is such a synaptic protein. So if this is present, basically a glioma cells formed a synapses with the surrounding brain cells. And you see, and it's also quantified here in the lower left, that upon FBOX knockout, we had less synapses of glioma cells with surrounding brain. And this is in line with actually a new emerging paradigm in the field of neuro-oncology. It's called cancer neuroscience, and it basically tackles the interaction of glioma cells with the healthy brain. And actually, we were involved in a previous study that was published in Cell this year, where also on a transcriptomic level, primary and recurrent glioblastoma samples have been profiled. And also here, the conclusion was that at recurrence, there is an upregulation of synaptic signaling processes. And again, in the infiltration zone, which is basically this lower right part here, there are a lot of proteins that are involved in synaptic formation. So this really seems to be very important in disease progression. And it was actually very nice that also on a proteomic level, we could confirm the relevance of this neuronal phenotype of recurrent glioblastoma. And this is basically one insight that we gained from proteomic data. But furthermore, we also built a basis for a new therapeutic approach. And this is shown here, and that's the second part of my presentation. So here, basically, in all the proteomic data from gliomas that we did, we found really a high abundance of extracellular matrix proteins. And there is an extracellular matrix protein that's called fibronectin. The counterpart is tennessine. and they were all at high level in our glioma proteomic data. So what we then did is basically based on literature research, it was very clear that there is a tumor-specific domain of this fibronectin extracellular matrix protein, and this is named EDB or extra domain B. And actually there is an antibody that targets or binds this tumor-specific domain of fibronectin. This antibody has the name L19. And based on other collaborations, we then developed a strategy for a targeted delivery of pro-inflammatory cytokines by using basically antibody cytokine fusion proteins. So the concept here is that we treat tumor-bearing mice or patients with these antibody cytokine fusion proteins. The antibody basically flows to the tumor site and brings the pro-inflammatory cytokines in a targeted fashion to the tumor. And of course, first we confirmed also this tumor-specific domain expression by immunofluorescence. And you see here, we have stained glioblastoma sections, but also sections from murine glioma models. And in green is this EDB domain of fibronectin. So it's highly expressed in human glioblastoma samples, but also in mouse glioma models. The beauty is that this epitope is species conserved. So we can use the same antibody for mouse studies that we also use for patients. And first we wanted to see if we can bring something in a targeted way to the tumor. So we used this L19 antibody, labeled it fluorescent, and injected it in orthotopic glioma-bearing mice. And basically what you see is that 24 hours after application, we had an accumulation of the fluorescent antibody at the tumor site. This was not the case if we used a fluorescently labeled antibody that cannot bind this specific epitope. So here we could not see basically something over the tumor site, and we confirmed this also by immunofluorescence. And then we used active payloads for this antibody that targets this highly expressed EDB domain of fibronectin. And you see here the survival curves, and basically we use different payloads, interleukin-2, human necrosis factor, or interleukin-12. And you see that basically all the active payloads conferred a survival benefit compared to the mice that were treated just with the antibody without a therapeutically active payload. There was no survival benefit if we again use this in an untargeted fashion. So this cytokines with an untargeted fashion did not confer a survival benefit. And we had an indication that we really trigger anti-tumor immunity because mice that survived basically this treatment were re-challenged, were resistant against another re-challenge. So they basically were long-term protected against another tumor implantation. We did a lot of work to find out how these cytokines work, but it is basically as you would expect. So they trigger local anti-tumor immunity, they increase the number of tumor-infiltrating immune cells, and particularly for the payload tumor necrosis factor alpha, we also found that this is directly cytotoxic. So this is basically this segmented area of necrotic, so meaning dead cancer cells, 24 hours after injection of these L19 TNF antibody in tumor-bearing mice. And you see in the control, the antibody without the payload, the tumor is intact. Whereas with the therapeutic active tumor necrosis factor alpha, there is really a dying necrotic area. And just to show that this is really relevant, it comes from proteomics, but it leads to a new therapy in patients. And we then translated this to patients with recurrent glioblastoma. So I mentioned before, these are patients where we have no gold standard yet. So this is room for experimental therapies. So we initiated basically a phase 1/2 clinical trial for patients with recurrent glioblastoma, where we investigated primarily the safety, but also preliminary efficacy of this antibody cytokine fusion protein. And this is some data of the first patients that we treated, or actually 20 patients in total. And the message is that it's a safe new treatment. So basically we had some flu-like reactions during the treatment, but no severe side effects. And the survival is not really earth-shaking, but it's encouraging. So in this patient population, you would expect a survival around 6 to 9 months. So we are slightly better, but still, this is not the solution for this deadly disease. At least we had an indication that we do something to the tumor. So glioblastoma is usually an immunological desert. There are not really a lot of immune cells. And at least what we could find are two observations. First, when we treat, we have a treatment-associated necrosis, so directly dying tumor cells in the center. This is what you also observed in the mouse data. And then when the patient progressed, we obtained their tissue and basically quantified immune cells. And yet you see that after the treatment, we really increase the abundance of tumor-infiltrating immune cells. And what we're currently doing now is to assess this treatment in combination with other chemotherapies. So in the second line treatment, we combine it with a chemotherapy that has the name lomustine, and we also combine it in the first line setting. And with lomustine, the data is really encouraging. So we can cure the majority of tumor-bearing mice. And again, we used proteomics to understand how this combination treatment of L19TNF and the chemotherapy lomustine or CCNU actually works. And essentially what we found is there are multiple mechanisms, but L19TNF really activates the tumor vasculature so that immune cells can better migrate into the tumor. And CCNU or lomustine, this chemotherapy, leads to DNA damage. And the combination of DNA damage and neoepitopes and immune cell migration really seems to trigger anti-tumor immunity. So this is the clinical trial program that we have, and that actually the basis for this comes from proteomics. So it is definitely clinically relevant. And just to show you that so far the data that we have is quite encouraging. So this is the first patient that we treated with a combination of this L19TNF and the chemotherapy lomustine. And it's a patient with a recurrent glioblastoma. And you basically see that in this patient, we observed a complete objective response of the tumor. So the tumor completely diminished. And so far, this is the progression-free survival of the first 9 patients that have been treated. It's quite encouraging because we have a median progression-free survival of several months. And if you go back to the literature, typically the median overall survival is around, the median progression-free survival is around 1.5 months or 6 weeks. So we are now better than the standard, and we're investigating this now in a multi-center setting. So to conclude, proteomic profiling is very powerful, and we used it to reveal novel insights into the evolution of glioblastoma. And we identified some proteins that are upregulated at recurrence, and especially we identified FBOX2 that seems to mediate the interaction of glioma cells with the microenvironment. In the proteomic data, we also find very abundant proteins that can be targets for novel immunotherapies or novel therapeutic approaches. And I just showed you one example of this fibronectin, the extracellular matrix protein, that we used for a novel immunotherapy with this so-called immunocytokines. And with this, I want to thank all the people involved, especially our collaboration partners at Guomics. And glioblastoma is a very challenging disease, but if we continue to study it, I'm quite optimistic we will find novel, gain a bit better understanding and find novel treatments in the future. With this, thanks a lot.
Hey, Tobias, I have a few burning questions.
Yes.
So you mentioned after the L19TNF treatments, you observed some changes in the blood, like ALT, AST increased. So there are selected enzymes representing the liver function. Have you done a comprehensive investigation of the serological molecular changes, like using proteomics? But then you can see the bigger picture.
Absolutely. We have not done this yet. We have the samples in the freezer, and it would be definitely very nice to test this and do this systematically in the future.
Okay, I'll be very interested to work with you in that aspect. So another question. So you mentioned a few combination. You said combination always works better than single treatments. But then you just say you combine with TMZ1R and CCNU lumustine. But how did you select these two options? You know there are so many potential combinations.
That is true. So far, this is a pretty boring rationale because this is more or less the standard of care. So TMZ irradiation is the standard in the first line setting. So it was very obvious to combine it because anyway, patients get it. And lomustine is, at least in Europe, the gold standard for patients with recurrent glioblastoma. So the rationale is just patients get it anyway. So we add it on top of the immunotherapy.
So the new therapy combined with the existing therapy. Oh, I see. I just want to say, since you, let's say if we have a global picture of the perturbations introduced by the new therapy, then we also know that introduced by the older therapy, probably we can better design the combination. There also the ratio, how much dosage, because if you have a two therapy at a full dosage, probably will introduce side effects.
Absolutely.
Synthetic lethality. In that case, both dosage can be reduced and you have a less toxicity to the patient.
Yes, we need the systematic studies, right, to have new combinations and better understanding. Absolutely agree.
So do you have a mouse model that you can test to see different combinations?
Yes, we have.
That we will be very interested to work with you to work out a systematic approach to test which is the best combination and which is the best dosage.
Yes, that would be really great. And this is definitely the future, to use novel combinations.
Cool. So any other panelists, do you have any burning questions?
Right. Actually, hi, Tobias.
Hi.
And this is Yifan. So a couple of things I'm not very clear. So initially you think that the recurrent glioblastoma don't change much with the initial primary ones, but it seems like the protein you found by proteomics is FBX02, which actually linked to a neuronal phenotype, and so the neuronal phenotype actually seems like much different. So how you think?
Yes, so absolutely, you are right. So let's say the short answer is molecularly, the tumors are not completely different. So based on genomics, transcriptomics, and also overall proteomic molecular signatures, the tumors do not differ much. But on a phenotypic level, so this neuronal phenotype, there is clearly a difference. And this is also what pathologists tell you. So they say at recurrence, the GBM is not really a mass, but it's really infiltrating. And it's basically a network with normal brain. So on a phenotypic level, there are strong differences. And it's actually very nice that at least we found a protein or a few protein that could mediate this phenotypic behavior. So the short message is molecularly not completely different, but on a phenotypic level, yes. So there might be a few molecular markers that determine these changes.
I see. So another thing is So as a patient selected, you select for treatment, are these patients actually you treat after the surgery? Or
Exactly. So basically, the first line setting is clear. So patients have surgery, so the majority of the tumor is removed, but there are always glioma cells remaining.
Exactly.
They evade basically the chemotherapy and the irradiation, and then the tumors can grow, have this more neuronal phenotype, and then there's another surgery, for example, and then a second line treatment. And this is where we try this new immunotherapeutic approach.
It actually make a lot of sense since as you said that the recurrent tumor actually looks like more infiltrative, and basically actually they come from the infiltrative or infiltrated aggregated cell already, and they come back. But this actually also bring another thing. Are the recurrent tumor in the recurrent tumor always, because when you do the surgery, it seems to me that a lot of blood vessel and they cut off, and the blood-brain barrier and more damage to them, and it seemed to me like it's more easy or recruit immune cells, right? So you basically bring a lot of blood in income and bring a lot of immune cells, and in the initial tumor, maybe less of them. And actually the phenotype you see.
Yes, that's a very interesting and important point. And actually, there is a current concept that you even do neoadjuvant. So you do some immunotherapy, you stimulate the immune system, but do surgery. And then the immunotherapy, when the tumor basically heals after the surgery, seems to work better. It's under debate. There are different visions in the field right now. But yes, this is a very important point.
And it also seems to me like if you do, and the antibody you link are like immune stimuli molecules. But your antibody like provide accurate and more enrichment of these molecules. Looks good, and thank you, and look forward to see the benefits in patients. Thank you, exciting. Thanks.
Thank you. Thank you.
Okay. Hey, Thomas, very good talks. Yes, like I have one very interesting question about the you talk about the first part of the recurrent gliomas. Like I one piece of data you have showed is like you have said there's a lot of genetic genes is involved in the recurrent part. So I don't see many much. I didn't see many piece of data like validation on this part. So what do you think of this part? Is it a specific like regulation for the like a recurrence of glioma by selected genetic genes?
Ooh, that's a very good question. I think. So ideally, what we would have is more longitudinal sampling, right? So far we have primary recurrent, but you are a surgeon, so I think in principle you could also, for let's say more low-grade tumors that have multiple resections, it would be actually very nice to have a much better longitudinal trajectory and to see actually what is really happening over time. And I think it would be also very important to understand what is related to treatments and what is the natural evolution of the disease. So far, this has not been addressed because, of course, all patients are treated in the first line setting, right? But I cannot tell you if the upregulation or changes in genes, transcriptomes, and proteomes is related to the treatment or is just a natural evolution of the disease. For this, it would be, of course, unethical to withhold the first-line treatment for patients. But somehow I think at some point this is very important and we need to address this. And probably we can start in really low-grade gliomas, probably where we have the time sometimes, right, to withhold treatment and then do surgery and get a better understanding.
Okay, thank you. Yeah.
Hi, Dr. Weiss.
Hi.
Hi. I also do research about brain. I draw the brain atlas, proton brain atlas, to show the functions of different brain regions. And I have a question. Many literature report that brain have most elevated protein compared with other organs of human body. And we do a lot of resources to show the different kinds of tumors. How is the brain tumor different from other type of tumors?
I'm sure Dr. Yang will answer and address this a little bit. So I don't want to give you a detail. So I think, so it's very, so at least based on my experience, based on the bulk proteomic data, it's not so easy actually to understand what is specific to the tumor cells and what is coming from the environment. And I think we need much more studies that assess this. So I think proteomics is a first step. But in a second step, you need the validation, ideally on imaging level or single cell level, to understand which proteins are specific to the tumor and which proteins are specific to the organ location. And this is at least not clear, at least from my point of view right now. And I think this will help to address your question. But probably Dr. Yang will cover a little bit how.
Have you ever compared with other tumor types, such like breast cancer or others? So
this is ongoing, but not systematic.
Okay.
Okay, maybe we go to the next speaker, Dr. Yang Hui. We can discuss later after Hui presents.
Yeah. For audience who didn't stay with us since the beginning, Dr. Yang Hui is a researcher, doctoral supervisor at Huashan Hospital Institute for Translational Brain Science at Fudan University. Welcome, Dr. Yang, to share your talk today.
Okay. Can everyone see my screen normally?
Yes.
Okay. Yeah. Today I will talk about like just because yes we have collaborated with Tiannan for long for 1-2 years, but the work is still going on. So I will talk a little bit about the our work from the proteomics in the glioma classification. And at the beginning, I think we I still want to make a story how why we need to do an omics study, like especially for proteomic studies in gliomas. Yes. So today, my part will focus on 2 parts. And the first one is like, okay. Oh, hello. What happens? Okay, sorry. Yeah, and the first one we are like to introduce the mechanism of very hotspot genes like IDH mutations in the gliomas. And the second part, and I want to like show some like a piece of data to introduce how we can learn from the IDH mutations from in gliomas. We need to introduce for the glioma classification from single genes to multi-omics. And so, if we so in the like past 1,000 years, I think in the earliest 50 years, we knew very little about gliomas. In the last 30 years, we have begun to gradually establish standard treatment protocols for glioma, like previous have showed for us like in previous slides. And also progress in the prolonging patient survival has been shown. Growing knowledges of gliomas enable us to remain optimistic about the breakthroughs in glioma treatments, like TTFields right now is widely used in the treatment of recurrent gliomas in children. So if we take the examples of the industry revolution in the field of glioma, we are still in the second part, and like the histology part. The actual part, and we know very little about the molecular biomarkers of gliomas. Before we talk about the gliomas, and I think we always want to show some data, just because IDH mutation, which is a hotspot mutation in gliomas, is a metabolic genes. So we want to talk about tumor metabolism. So it's a long time story showed here. Actually, when the normal cell becomes to a tumor cell, they adapt the metabolic pathways very quickly. And this phenomenon was called Warburg effect, was discovered by Otto Warburg in 1920s. And actually, in a few weeks, in a few words, the Warburg effect is like the tumor cell replace the respirations of oxygen in normal cells by fragmentations of sugars. They use a lot of glucose, but the generation of ATP is not equal to the consumption of the glucose. Also, the underlying mechanism of this effect was not remain unclear. However, the application of this effect has been widely used in the diagnosis of tumors like PET-CT, and technologies. So in 2008, I think this is the most important discoveries in glioma, and I think it's also for tumors. They it's the next generation sequence and found the first metabolic genes is IDH was mutated in GBM patients. This is the first time, and this is the first time this is the first time for human to discover the metabolic genes is actually mutated in GBM patients, and this is I think is a very important clues that links the metabolic regulation in tumor developments. After that, I think a lot of groups just joined to sequence more samples, and finally, I think found the IDH mutation account for approximately 80% of lower grade gliomas. And in young adults with IDH mutations, radiotherapy or surgery existing can be beneficial, and is shown here. So it's very special right now. And one famous researcher is called Yan Hai, and he also started a company in China right now to for the diagnosis of gliomas. And it's New England Journal of Medicine paper. They found the IDH mutation. The patient with IDH mutations showed a longer survival rate, and this is the first time link one molecule with tumor survivors. So after the discovery of the IDH mutation, and at the time I just graduate students. So our lab and at the same time, it's very interested in the mechanism of IDH mutation, how it causes the tumor formations like gliomas. So the first time when we express the tumor-derived IDH mutations in the cells, we can see the activities, the normal activities for IDH that confers like isocitrate to FKG is totally abolished. However, 1 year later, other group from carrying and found the mutant IDH gate other function, it can convert the FKG to other metabolites called 2AG, which is highly accumulated in the patient. So after that, in the lab, we just established the metabolites detecting of several metabolites like FKG and 2AG. We also found the patient-derived mutation of IDH1 can cause the 2AG accumulation is as high as 1,000 times when compared to the wild type 1. And we also found the structure of 2AG is similarity to the FKG. So at the time we have a hypothesis that FKG and 2AG can share, like 2AG can serve as a competitive inhibitor for the FKG dependent some enzymes. As we know, there's many kinds of enzymes in the cells that use FKG as a cofactor, like histone demethylase or DNA demethylase TET families. And for shortened like long story, we have found that the accumulation of 2AG can inhibit a range of dioxinases, and which lead to the like DNA histone methylation changes and relate the gene expression, and ultimately promoting the tumorigenesis. And so this is the first part. We have found the IDH mutation, which is important and connect the tumorigenesis from metabolic part to the epigenetic part. But when we did these studies, we have talked more with the surgery of Huashan Hospital, and we know from the clinical, the only one IDH mutation cannot do the classification for all the tumors. So what we want to know is like what we can learn from the IDH mutations. We have did some like translational studies. So during to the time course, we will not show it here today. So as we know, I think Tibia also mentioned that the heterogeneity is one of the most important characterizing gliomas. So we need to find out more molecules, subgroups under the single biomarkers to diagnostic scales. And very recently, we reviewed like thousands of samples in Huashan Hospital. We found like if we include more molecules like IDH mutation and TET mutations and EP19Q deletion or EGFR amplification and the BRAF mutation and H3 mutations. If we include 6 molecules, we can classify the tumors in. We can classify the tumor cell into 6 subgroups, subtypes with significant difference of the survival rates. So this is very important, and it tells us maybe we need to change it to like a classification of the tumors from one genes to many other genes. And also from here we can this 6 molecules types has a completely different clinical diagnosis, and the results also demonstrate the molecular marker-based grading is more acute for the progression and like WHO gradings. In our study system, patients with WHO class 3 and molecular grade 1 tumors have better progress than WHO class 2. Molecular grade 6 tumors. Based on this, we have also established a flow chart for molecular integration of multiple molecular markers for dioxins. So right now, and we know this picture is with the continuous development and genomic studies, I think we have increased our understanding of this molecular biology of gliomas. The combination of individual molecular markers alone becomes insufficient, and we must move faster into the fourth industrial revolution. That's using proteomics, especially for proteomics, to make more acute glioma dioxins. So like this picture, I think a lot of people know it. Yeah. If we, I think for now, if you use single gene dioxins, it's just tantamount to a blind man feeding the elephants. So right now we need to use omics studies to find the whole part, the whole characteristics of the elephants, so we can make a acute dioxins for these pictures. So at present, I think mostly studies are a reference to the TCGA, and studies is dominant for a long time in the United States, and very recently and in Germany, and they have presented to use the methylation analysis, and which can complementary to both glioma and meningeal gliomas. So with the completion of full sequence of human genome, life science research has gradually entered to the post genome area. And with collaboration with GoOmics, and we have started to use proteomics to diagnose the gliomas, especially for the high throughput proteomics technologies, which is well established in Go's lab. So use this method, we can identify as many as molecules that can affect the pathology states as possible, and so this will gradually becomes one of the core elements for biomedical research. The benefit of proteomics has been demonstrated in sequence for variety of intracranial tumors in meningeal gliomas, and this paper, and as published just last year, found the first time found the meningeal gliomas can be classified to 4 subgroups with different survivors here. And this is the first time use multi-omics data to classify gliomas. And more important, by using this method, and they continue to use the proteomics data to generate the molecular markers, especially for the protein markers for each subgroups. There are many markers, protein markers. At last, they choose S100B and SCNG, ACADL, or MCM2 for the MG group 1, group 2, group 3, and group 4. And importantly, they can use a simple IGC to validate this dioxin of each group. This study uses proteomics will provide a new tools to us to identify new markers, and can be used used by IGC to identify the different subgroups in the tumors. And so, and in the last year, and I think it's the glass I don't remember clear. Maybe it's the glass group. They have like integrated the protein genomic analysis of the glioblastomas, and including metabolic and lipidomics, and also single nuclear RNA-seq. They have found that the glioblastoma can be classified into different subgroups with immune markers. So this study have provided the new therapy opportunities for each type of gliomas. So if we and the other things is like always we have been asked why we study proteomics. Other studies also published in last year. We have read many times. They have tell us a story. Tell us answers is when we combine the proteomic data with the transcripting data, and there are a lot of always the same. There will not always the same in one sample as compared to other one. So if what this paper found, they found they can use some like RNA genes and the genes of RNA levels to classify the glioma, white type IDH white type glioblastomas with different survivors. However, if we apply these genes to the protein levels, they didn't find any significance. So in the protein level, and they have generated a protein specific protein specific the tumor subtypes by using the proteomics data. So this study and for the first time told us maybe the protein will be the better marker to classify the gliomas when compared to the RNA in the omics data. So in our samples, we have also did multiple omics studies before we start the proteomics studies. We also want to answer one question is after we found the IDH mutations in gliomas, so we want to use some other metabolic molecules, whether we can classify the glioma subtypes not only by the 2AG or some other molecules. So we did the unbiased metabolic studies. We have also found the IDH mutation actually shows more significant difference from the IDH wild type one with several kinds of metabolic molecules. So this data told us use omics studies, we can classify it very clearly with the genes involved in the metabolic with the genes mutations involved in the metabolic pathways. So later we have also started the proteomics studies. So due to the studies going on, so we will show part of data to propose what we have found. And use unbiased studies here, we can see we also can classify the samples, the patients with three groups, and significantly, and with group 1 and group 2 shows better survival, and group 3 shows a worse survival. And all these genes involved here is only for the metabolic genes we used, and we can do a very clearly classification of this tumors. This is more than 100 samples we used. And also what we want to know is as we have mentioned, the IDH mutation is not a good marker with a single gene to classify the whole glioma. So we choose the IDH wild type 1 gliomas out from the samples. So if we do the unbiased studies, we can see there's a very significant changes between the there's very significant changes in two groups for the proteins expressions. If we choose to check the survival of each group, we can see group 1 or group 2 shows a significant difference. what this means is like use the proteomics data, we can still reclassify the single genes subgroup of gliomas. So due to the studies, I will stop here right now. So this is a very interesting picture. Is drawn by the director of our department, Zhou Liangfu Yuan. And usually in the clinical, and the most important question is how we can find the tumor acutely, and then then cut to fight the tumor. So the tumors need to be more people involved to study the glioma to find the tumor edge or some tumor markers to classify the different type of tumors. So lastly, I will thank all the people in the lab, and also the doctors Mao Ying and Liang Hao from Hua-Song Hospital, and also Tiannan from Westlake University. Thank you.
Thank you. Very interesting presentation, and also the drawing, the cartoon is really interesting. So any questions? So let me ask the first question. So for the proteomic pattern that you showed in the unpublished data, so how do you think this? Do you think this has a clinical implication? Will this be possibly applied in clinic? These are, if you look at these different groups, 1, 2, 3, there are so many proteins are different, some up and some down. Do you think we can use this potentially in the clinic to stratify the patients?
Yes, this is very important question. I think this is what we want. I think it's important. This is why it's important to use proteomics to study this classification of gliomas. And I have mentioned one story here. It's about the meningioma. Is also like when you use the transcription, also isolation and omics data to study and classify these tumors in 4 groups. So finally, they still use the proteomics data to find the biomarkers to identify different groups. Usually, if you use proteomics, you can find the antibody. You can do the IgG, which is very simple and cheap for the diagnosis. So I think we have the same arms, like we want to use the proteomics data to try to find several protein markers, and then we can develop some like IgG assay to identify different groups, and finally can use it. I think it will be can use for the clinical part. Yeah.
So in the introduction, you showed one paper. I remember it's saying combination of single gene mutations can stratify glioblastoma. Because I think most clinicians, they want to see a single gene or single protein, which can be binary classified, let's say IDH mutated or not mutated. And if it's mutated, then it has a response to certain therapy, but if not, then it's resistant. But from this heat map, you will see group 1, 2, 3, they have so many proteins, they are different. It's not a single gene event difference. It's not only a single protein. So in clinic, do you think the clinician wants to see among this 100 or 1,000 differentially expressed protein, do you want a single gene or single protein classifier, or do you think it's also acceptable to use a multi-protein classifier? for this classification?
Yeah, it's a very good question. Yes, also. So I think for the clinical use, we want to develop as simple as possible to detect the tumors. Use simple markers. If we use hundreds one, I think no one wants to use it. So for the proteomics study, yes, actually we have found many markers. I think hundreds of these markers, I think is the important one. We want to use the computer to calculate out the most important one, like choose 10 or even less than 10 markers to classify. I think this may be more important if we stand by the clinical part. Yeah. So the research study, I think if we include more, we can see different pathways, just like previously actually shows selectogenic pathways important for the recurrent part. Actually, in our study, we also find the selectogenic pathway is important for glioma group, like for the glioma progressions. I didn't show the data today, so we didn't know the reason, but this kind of omics data will provide a big picture for the studies. But finally, we still want to use the different computer to calculate it out, give us only 10 or even less 10 markers for different subgroups. Yeah.
In addition to stratification, do you also have the chance to find new therapeutic targets which you can use eventually to cure some group or all of this glioblastoma? Have you checked the druggable protein in this list?
Yes, this is. I think this is the work we are still going on. Actually, in the morning we have a meeting and we have found I have seen some positive feedback from students. They have found several metabolic genes and involved in the different group. If we look at each of them, and we can see very significant prolonged the survival for in the in vivo mouse models. So if we look at other ones, which will not have benefit in some of the types. So all the work was done in the patient-derived glioma cells. So that's where a plus in this study. So I think if we add this part, and this will attract more and more people to studies like the new protein. I think proteomics data always give us a solid part. We choose the several genes is can be easily validated from this omics studies. If you choose from the RNA-seq data, sometimes it lost work. I think this is why this is different from RNA data and protein data.
Actually, it would be very nice, just a question, to understand the reproducibility of the multi-protein signatures of the subgroups. So for example, if somewhere in Europe, we also do proteomics with a different mass spec, of course, we probably won't identify the same proteins, but at least it would be very interesting to see if, let's say, we identify, I don't know, 50 out of 200 proteins that you identify are specific for each subgroup. If we also can separate the different subgroups, right, by a more restricted multi-protein profile. This would be actually, if this would be the case, this would have huge impact for future classification.
Yes, it's a good suggestion here. And actually, we have read several kinds of several papers about the proteomic studies for gliomas, and we did also use the unbiased study for all whole proteins, but this will give us even more information. I don't think it's is better than right now we choose a small piece of them. Use the whole pictures of the genes, the proteins to do the unbiased studies. We do find some like pathways which is important for the progression of the gliomas, which is also associated with the different survival rate and in this data, but we cannot find some like very important markers just because hundreds of genes like is involved in this part. So so we want to like study from another part like a small subgroup of proteins like metabolic genes or immunologic genes to study the changes during this gliomagenesis. So from our data, and we also to check the upregulated genes or downregulated genes. In the upregulated ones, we found several of the metabolic pathways. The genes involved in the metabolic pathways is published by some of the papers before. So I think the data from our samples is a validation and to that part of papers. So but there's also some new genes is involved in different pathways, which is the first time identified by our data. So we are just like validating the use of patient-derived cells to see whether these genes, this proteins is involved in the progression of gliomas. And actually, it's from data we have I've seen in the morning, and it is yes, yeah.
Look. Right.
Yeah, please.
Look like so many exciting to see so many data you generated. So have you compared your proteomics data, things like Tobias with just said, and the genomic classification looks like clear and they used for many years, but it really doesn't apply to patients. This means actually really we need a new technique and new ways to classify them. But by the way, do you compare these data to you have done with the IDH subtype, but do you compare these with known classifications, and do they really have huge difference?
Okay, well, what compared to what?
The genomic classification of the glioma.
Okay, yeah, actually, this is a really important part of what we want to make. Yes, from the cell report papers, they found the difference. They have found many differences, like from proteomic data to the transcriptomics data. So in our data, we also found the proteomics like protein levels. If you use this omics data to classify the subgroups, and we also found some examples, like can find some of the if you use the unbiased studies, this is from metabolic genes, we can see some like high-grade tumors here, but not all. Some of them is classified in the low-grade, more likely the low-grade ones, which is we have found in the clinical that some of the GBM patients actually will be survival with short survivals. However, some of them survive very long. So I think this is if you use the classifications is not acute to diagnose these gliomas. So for the IDH one, what I showed here is actually I also want to mention that not all IDH wild type gliomas like shorter survivals compared to IDH mutant ones. So we want to use the proteomics data to reclassify this subgroup, this group, to show whether we have difference, and we want to tell how the proteins involved in the IDH-white gliomas, which affects the survivors. So actually, from this data, we can see significant changes between IDH-white type gliomas, and then they can classify it into two groups. Yeah.
Mm-hmm. By the way,
yeah.
How many examples you already done? Seems there's so many.
Yeah, it's I think 180.
Wow.
Yes.
I
think it's a lot of data. Yeah.
Right.
But we didn't have the recurrent tumors. Yeah, in your samples, we only have the primary ones. Yeah, that's.
You only have primary one, and
yeah, we don't have recurrence. Yeah.
So then another project, and
yeah, this is a very precious resource. Yeah, go ahead.
Right, exactly. By the way, do you see any like some secret proteins? And it seemed to me like these, if certain secret proteins identified, and that maybe then we can diagnose this without getting samples. We just get some fluid on there.
Yeah, that's a good idea. Yeah, we haven't done it now. Yeah, I focus on the metabolic part and also the immunology part. Yeah. It's a lot of work needed to be done. I think this data is generated by Tiannan. I think it's like a book. We cannot we cannot.
Can you share with me? We dig it together.
Yeah, Tiannan has a very good technology to detect even more proteins when compared to other groups. We use the mass specs. Yeah, so that's very important. I think. Yeah, I hope we can find something new. Yeah.
I think that's because the cancer and the tumor and all this cancer are very heterogeneous, and they have so many molecular changes. And previously, without this technology, we cannot see them. They are there, they're always there, but we could not see them. Now we have technology to uncover these changes. There's so many differences. And that's the reality.
Don't have meaningful ones, especially.
Yeah, I have a question to Hui. So compared to the cell line study and the tumor, the mouse model study, to analyze the tumor from the patient, you get the information not only from tumor cells, but also some other cell types or secret proteins in the microenvironment. And now there are so many therapies against the immune system, and maybe combined with chemotherapy, radiation, and so on. So from these proteins that you identified, which are different expressed, do you, just now you mentioned the metabolic enzymes. I guess those are from tumor cells because you are talking about the Warburg effect. But do you also see differences from the microenvironment? Do they have indication for therapy or diagnosis?
Okay, this is a good question. Yes. Actually, we also want to study the microenvironment, which Tim also mentioned. Yes, it's also very important and in gliomas. So for this work, what we have done, like when we collect the samples, we have choose the core regions of the tumors, and with a very near piece of samples. So I think Tiannan was low. So I don't think in this piece, in the little piece of samples, they include much more the lung tumor cells. So from the proteomic studies, we do find some changes. I think most of changes are from the tumor cells. If we want to study the microenvironment change, so maybe we can develop the single cell like proteomics, and we are interested in. Yeah. So right now, most of people use the single cell RNA sequence or single cell nuclear sequence to study the environment change during the glycogenesis. And also compared to different subgroups, they do find some markers from the immune cells or the your surface, the member surface markers. Yeah. So from proteomics data, I don't think we can identify this kind of markers which can be used for diagnosis or therapy opportunities.
Okay, thank you. So any other more questions? Chelsea, do you have questions? Sorry.
Oh, hi. Yes, I have a question. So both Dr. Weiss and Dr. Yang do not mention about the originating of the tumors. The tumors originating from spinal cord or from the brain cortex, are these have different clinical and molecular phenotypes?
Oh, I say first, Tibet.
Yeah, go ahead.
Okay. Yeah. This is a very important question. We have missed. I think during the analysis. Yeah, I think most of the tumors right now we use is from the cortex. Some of them is close to the surface of the to the surface of the brain. But I think about 90% of the sample is from the cortex. From the clinical, they do found the localization of the glioma affect the survival rate. So maybe we can include this information in the next analyzed. Yeah, it's a good suggestion to us right now.
And just as an addition, actually the location matter a lot. So it's known that, for example, if the tumor is more in the midline, it has special histone mutations. It's rather at the edge of the cortex, it has another histone mutation. If it's in the parenchyma, it has a different. So this is known from genomics and transcriptomics, but the location matters a lot.
Yeah, as I know also, the location will affect the growth factor used, like EGFR, interleukin, insulin receptor, and MET and others, and it's to have a lot of difference. You can refer to two papers. One is actually from Nature recently, another in Zhejiang University. Another is from MSK.
Yeah, please go ahead. Sorry for the echo.
I mean, Xiaoqi have a question for the location, the relationship of the location, tumor location, and its outcome or phenotypic relevance, and so the other couple papers. I know two, at least, and one is from Zhejiang, actually close to you. Another is MSK, Luis Prada's lab, and they have a cancer cell paper talking about the location and the different phenotypic difference of the tumors.
Thank you.
Dr. Guo, I'm sorry, I think you're muted.
Oh, sorry, I was muted.
Hello, I'm Ray. Hi, Prof. Yang. There's a question from the audience. Could this approach distinguish different stages of tumor?
Oh, you mean use the proteomics data?
Yeah.
Yeah, of course, it can distinguish. So that's why we do reanalyze the samples. We just divide the samples in two parts, which the one part is from the grade 2 or grade 3, which also low as the low grade gliomas, and the second part is grade 4, which is GBM, have different survival rate by significant difference. So I think that's if you use the protein classification, they will be classified in two groups. So in order to find the different markers, we just choose like this to study in the subgroups of these two kinds. So we maybe find some real factors that affect the grade. Maybe some of them high expressing in grade 2 or high expressing in grade 3. If we use the whole samples, I don't think we can find. Yeah, we only can find some proteins highly expressing in grade 4, but we cannot see the difference between grade 2 and grade 3.
Yeah, thank you.
Okay, thank you. I think we already running out of time. Thanks for Tobias and Hui for this very interesting discussion and presentation. I think glioma is definitely a very lethal disease, and they occurred in different regions of the brain, probably have difference, and they have also very complex microenvironments. This is really understudied. And as Toby just mentioned, you tried a new therapy and combined with older therapy. I remember he used the word boring because there are not much options available in the clinic. And the reason behind this is we know very little about the biology behind this disease. There are so many molecular changes that as shown by Hui's data and by Tobias' data, by proteomics, we can see so many protein difference. Some from the tumor cell, some from the microenvironment, and some from probably because of the tumor region difference. This is also understudied topic, but we need to have a large volume data before we can systematically analyze this. It's still a long way to go. And I'm also curious about the difference between the patient in Switzerland and China. Probably there are also the population difference, and it's also understudied. Most of the clinical trial and medical guideline, they are based on the patients in the Western countries, and they are directly applied in patients in China, whether there are any difference and what are the differences, and this also understudied. I think today is a very good opportunity for us for the young scientists to have a communication. I think this is only a start. Maybe in the future we can compare the proteomic data from Tobias patients to Hui's patients. They are quite a large amount. Probably can find something more interesting in the future. And thanks a lot for the audience. Thank Professor Su, thank Xiaoqi, Sun Rui, and I think that's it. Okay, then have a nice day. This is the end of the symposium. Yeah.
Okay, thank you.
Thank you very much.
Thank you. Bye-bye.
Bye.
Bye-bye.
Bye-bye.